Okay, so let's kind of get into just the mechanics of cell division here. Let me find my notes. Now you have nice figures kind of showing you illustrations next to real pictures of cells in mitosis. When you get to the microscopes, I mentioned this before, you can view cells pretty well at 400x. For us in this class, that'll be our high mag. If I ever say a high mag, the total magnification. And we have a medium and low mag. There's like also 140. Those are the three magnifications we'll use in today's lab. You at least got to get to 400 so you can begin to see some stages of cells and some stages of mitosis in the slide you got for that. Anyways, I think when I take students do this, I just kind of make a table of what you should kind of pay attention to as you're studying these figures. Note the phase of mitosis, mitotic phase. Note the mitotic phase. Note, what the DNA is doing. Yeah, how does it appear? What's going on with it? And realize that we're packaging the DNA from moving around to be divided into two cells, and we call it the chromosomes. Okay, the genetic material. Another word. I mean, it's like, they call them that. They could see them under the microscope many decades ago. They're very colorful. So chromosome is a derivation of the word chromatic, which means color. We know now the genetic material is DNA. I mean, for a while they didn't know. They thought, they always use the term genes. You heard the term genes? It's in your genes. They didn't know what genes were. They didn't know they were DNA. They could have been carbohydrates. It could have been proteins. Turns out it's DNA in the chromosomes. So pay attention to that in particular. Also, the nuclear envelope, they have their own membrane. Either it's going to be there or it's going to dissolve so you can move things around. And also, the spindle apparatus is a good clue. The spindle apparatus are the two centrosomes. They make kind of a spindle in certain phases. So this figure has like the first three phases as it goes through. And I wish this were an animation instead of freeze frame, but maybe you could look up an animation. Let's kind of just look at each phase individually. The first phase isn't even a mitotic phase, okay? It's interphase. So I'll put it here under mitotic phase, but no, in parentheses, because it's not part of mitosis, but it's right before it begins. And the thing you could tell if it's interphase about to be mitosis, it'll be like super late. I'm going to skip to spindle apparatus. Notice you won't have one, you'll have two centrosomes and they're close together. That's kind of usually the clue. Two centrosomes close together. That's usually how I tell I'm in late interphase. Because what's going to happen is those two centrosomes, they're going to move apart. And when you're done with the whole thing, each daughter cell will have one centrosome, which is what a cell normally has. You don't normally have two. So two is the clue. Also pay attention to the DNA. It starts to condense. like you're coiling it up, you're packing it up. So that when you move the chromosomes around, you don't damage the molecules. Okay, kind of like when you pack to move, literally move from your apartment to a house. You pack it carefully, and if you don't, stuff breaks. The nuclear envelope is still, it's fully intact, but you have to break it down. It's just not broken down yet. I'll just put, it's still there, it's intact. When we start to go through the phases of cell division, start with, they call this an early prophase. I'll just say that the DNA, it's already replicated, okay? It starts to condense more. Remember, when did replication happen? It happened in interphase during the S phase. That's why they draw, well, DNA chromosomes normally are drawn like this. That's one set of chromosomes, one pair. You got one from your mom, one from your dad. That's your inheritance, whoever your biological parents are. Paternal, maternal inheritance. When you divide a cell, you have to replicate that. We already did it in the S phase. I just didn't really mention this detail, but you just make a carbon copy. So I had a pair, now I have 4. It's still a pair, it's just each part of the pair is now replicated. And that's what they're showing you there, in case you're wondering. Now notice this term, centromere. The 2 chromosomes that are copies of each other, these 2 purple ones, maybe they're from mom. They're attached at the centromere, and that's the detail you should know. So I'll draw this really big. There's a small part of that centromere. Okay, this centromere is not the genetic material, it's just kind of holding them together. But there's a small part where the microtubule will attach. I'll kind of color it a different color so it stands out. I'll put a little green part there, a little green part there. That's called the kinetochore. Let me spell that out. Yet another word to add to your list. That's where the microtubules will attach to move the chromosomes around. So that's what you got there. Okay, replicated chromosomes, they're condensing. And also look at the nuclear envelope. Still there. It's going to break apart, but it's pretty much still there. I mean, that's how they illustrate it anyway, so I'll go along with that. And also, the 2 centromeres are not close together. They're separating. Okay, so I'll just kind of put separating. So you go from early prophase to a later stage of prophase. You can kind of see some things develop here. All right, so now we've got late prophase. By now, you've got both centrosomes at polar ends of the cell. They're at opposite ends. And we have what's called, this, well, they call it a spindle pole. It looks like a spindle shape. That's how they describe it. I'll put that. You have spindle pole. The nuclear envelope is pretty much gone. It's broken down. It says fragments of nuclear envelopes. I'll just say broken down. The chromosomes, what's happening is microtubules are attaching to the kinetochore. They're going to move them around. The goal is to move them all to like the center, the equatorial plate. And that's, when it gets to that point, it's called something else, metaphase, okay? Pretty much the chromosomes, they're very condensed. It's very easy to see the chromosomes. And they've been moved, they align at the equatorial plate. Okay, they call it metaphase plate, so I'll go with that. Chromosomes line up at metaphase plate. Nuclear envelope, still gone. Okay, it hasn't reappeared yet. And the spindle apparatus, well, we just say this is kind of, you're still in full spindle mode. Okay, it looks like that spindle. So you can see what's going to happen, or maybe you don't. It's like, why did you line them up? So you can pull them apart. Because remember, the DNA was already replicated. So you don't want it replicated when you're done. You want it not replicated. That would be the proper amount of genetic material. So, you know, you just pull them apart, and that's shown here. We can call that anaphase. So you have the characteristic kind of pulling the chromosomes apart. They call them daughter chromosomes. They're just chromosomes. They were carbon copies of each other. They're literally copies of each other. Can't use the term carbon copies. Young people know what that is. It's like when you cut and paste. What did you paste? Well, shouldn't it be the exact thing you copied? That's what this is. This is a copy of that. When you pull them apart, you got two chromosomes. Maybe they made some switches of some genes. But technically, when they were copied, when they were replicated, they're the same. So when this cell and this cell will have your regular genetic material. So you separate, I'll just say separate daughter chromosomes. Now this is when they separate, but when they're together, just to avoid confusion, technically your book may call them, when they're just two chromosomes, but people call them different things. When they're attached, they call these two replicated things sister chromatids to each other. And when you pull them apart, I guess they call the sisters daughters. Sometimes I say separate sister chromatids. I don't want you to be confused. You're just pulling the two chromosomes apart, regardless if they call it a daughter or sister. Okay? The terminology doesn't matter. It matters you understand what's happening. So if I say separate daughter chromosomes or separate sister chromatids, same difference. Don't be confused by what it's called. The nuclear envelope still gone. You're still in spindle mode. Okay, it's just kind of a full spindle. You got to finish separating the chromosomes. You can start to like pinch off the cells there. After anaphases, telophase / cytokinesis. Telophase, cytokinesis, shown here nicely. Now telophase, pretty much you'll start to see the nuclear envelope reforming. They say forming, I'll say reforming because that's the form again. So you finish moving the chromosomes so you can unpack. You can start to, I don't know, uncoil, decoil. I mean, you got to do that, otherwise you can't access the genes. You can't turn them on. So just like I gave the moving example, once you're done moving, we have to unpack. I guess you could live out of your boxes, but that's really inconvenient. You can't get to anything. So you uncoil the DNA. The spindle's gone, because now what you got is each cell, as you can see, as it starts to divide, one centrosome in this is going to be a daughter cell, and the same thing here. So there's no spindle anymore. Okay, you're starting to pinch off right in the middle. Now, telophase / cytokinesis are kind of happening at the same time. I mean, so maybe it's best to not include cytokinesis as part of mitosis. Cytokinesis is an equal division, sometimes it's unequal. In this case, it's equal division of the cytoplasm. The cytoplasm has everything, all the other cell organelles, which aren't illustrated. But you want to divide those in a way where both cells can be functional. So I'll say equal division of cytoplasm. So for example, if you have one cell and it goes through mitosis, how many cells are you going to get? 2. That wasn't a hard question. And if these 2 divide, how many are you going to get? 4. And really, each of the 4 should be the same as the top one. The only example you'll learn that's not equal division is the oogenesis, the egg, in female repro. That's unequal. It's like, instead of like dividing equal, it's like, it kind of divides like that. That one, a little one's going to pinch off, and it's like, got one big mama egg. And that's going to divide again. You're going to get like, that'll divide, that'll divide. So in female repro, it's like the 4 cells, the 3 are called polar bodies and they're non-functional. The egg, that is what's ovulated and fertilized by sperms. That's the only example where it's like, from the 4, you only got 1 that you need. You ovulate 1 time per month. Well, you learn about that. It's not always equal is my point, okay? Now, where you see the pinch, it's called cleavage furrow. Look for that. Look for that's kind of where it's pinching off. This is happening during telophase, so they kind of happen at the same time, okay? They occur during telophase. that'll conclude lecture for today. I want to get you ready for lab. But let me present to you the cells I want you to look at under the microscope. So shifting gears towards lab, get out your printout. I'll walk you through it. Take me another 20-30 minutes, and that'll leave you plenty of time. Okay, class ends at 11:45. This lab doesn't take long. It's pretty light in terms of labs. Oh, okay. I did make a few extra copies, so I can put them out there. Either bring one, but you can work alone or in partners. This lab is due today by the end of class. All of you need it? I don't have enough. I'll make more copies.
We can share, so I'll
Okay.
just share it.
Okay. Okay, I'll make copies for you. I need this. Don't worry about it. Did I say to print it out? I think I did. Yeah, you're saying I did, so I guess. But you know, get in the habit of doing that. We won't always have a lab to print out, but today we did. I understand it's only the second day, so you're not quite with it yet, but you'll find your sea legs soon. So I'm not going to explain the whole thing, but read through page 1 and be careful. The way you lose points for lab are leaving things blank. Do you see the first blank on the first page? What number are you supposed to fill in for that blank on the first page besides your name? That, I want to know what scope you use, so if I get it out the next time, it's all messed up. I have a record. But what about the bottom of the page? Where are you supposed to put? Number 6, ocular lens, magnification. What is the magnification of this lens? I'm just going to tell you some of the things so you're not frustrated trying to look it up. Let me clear the board. All right, this is where I can stop this. I don't need to record this part.